My name is Kevin Harlan. I'm a scientist at Montana Molecular, and today I'd like to tell you about some of our recent advances we've made in using the biosensors and assays that we create at Montana Molecular to study things like cell stress and toxicity in iPSC-derived cell types. In this presentation, I'm going to be focusing on the use of our assays in iPSC-derived cardiomyocytes. Cardiotoxicity is a side effect known for many different drugs, especially chemotherapeutics. It's also been established that cardiomyocytes are very sensitive to these different compounds. And as you can see on the left here, when we treat either HEK cells or core-for-you cardiomyocytes from Nkardia with the chemotherapeutic doxorubicin, we can see that HEK cells are insensitive to this treatment, while cardiomyocytes are highly sensitive. Thus, it's very important to use these iPSC-derived cardiomyocytes, like the ones here from Nkardia, for use in screening for cardiotoxicity. Now, we have these great iPSC-derived cell types, but we also need assays that are capable of being utilized in all these different specific iPSC-derived lines. And that's where Montana Molecular comes in. We've been working to develop our assays specifically for use in iPSC-derived cell types. So for instance, we can use our assays in iPSC-derived peripheral neurons, as you see on the left, or in iPSC-derived cardiomyocytes, as you see on the right. Importantly, our assays can also be detected on a number of different fluorescent detection schemes, such as standard plate readers, imaging-based analysis through microscopy, or even high-content analysis through imaging-based plate readers. Today, I'll be telling you about the use of our newly developed stress biosensor for use in core-for-you cardiomyocytes and screening for cardiotoxicity. The stress sensor is capable of detecting mediated cell stress. On the left, you'll see unstressed cells are marked by a red fluorescent nuclear localized protein. If we induce stress by adding an stressing compound, we induce a green fluorescence, which can be seen on the right. Using this assay, along with our assay for calcium, known as rGecko, we decided to screen for different cardiotoxic effects of a number of different chemotherapeutic compounds, including doxorubicin, tyrosine kinase inhibitors, and another class of cardiotoxic compounds known as cardiac glycosides. To do this, we plated cardiomyocytes in 96 well plates, transduced the cells with either the stress sensor or the calcium sensor, added these different chemotherapeutic compounds, and measured stress at 24 hours or calcium fluctuations and beating at 8 and 24 hours. We first quantified cell stress using a method that detects the percentage of green or stress cells within a population of cells that have been transduced with the stress sensor. And what we can see is that if we look at the increase in the percentage of stress cells upon treatment with each of these different compounds, we can see that the cardiac glycosides, digoxin and digitoxin, increase cell stress at lower concentrations, along with one of the tyrosine kinase inhibitors, sunitinib. Looking at a higher dose, we again see digoxin and digitoxin produce these increases in cell stress. We also see now some of these chemotherapeutics, such as doxorubicin and erlotinib, also begin to produce cell stress. We also looked at the calcium dynamics and beating rate in cells treated with all of these compounds after 24 hours using our rGecko calcium sensor. And we can see that cells treated with DMSO have a nice coordinated beating, similar to those treated with doxorubicin. However, our beat rate and amplitude has slowed. Similarly, we see a slowed rate, although no change in amplitude in cells treated with lapatinib. And in those treated with digitoxin, we basically see a full loss of coordinated beating. We can then compare the changes in cell stress, as well as changes in beating and calcium dynamics induced by each of these different compounds, and ask which compounds are more similar to one another and which are more different. Here, what we've done is create a correlation matrix where compounds that are more similar to one another are in blue and compounds that are more different are in red. And what you can see is we've really identified three different classes of compounds. Here we have the cardiac glycosides clustered together, and then we have two of the different tyrosine kinase inhibitor compounds clustered. If we compare this to a known cardiac safety index, comparing all of these different compounds together, it matches up quite nicely, where we see doxorubicin and sorafenib seem to be the most cardiotoxic compounds, followed by lapatinib and sunitinib. And what these data have shown is that we can use these different assays from Montana Molecular, in this case the ones for cell stress and calcium, to analyze cardiotoxic side effects in iPSC-derived cell types, in this case iPSC-derived cardiomyocytes. For more information on these assays, please visit montanamolecular.com.